What if I have a quaternary protein, over 400 amino acids in sequence, made of 2 alpha chains and 2 beta chains. Each of these more than 100 amino acids in sequence. And on the beta chains, on the 2 beta chains, I make a mistake in 1 amino acid at the 6th position. I substitute a polar for a nonpolar amino acid at the 6th spot. It is actually a glycine for aspartate substitution. Glycine's supposed to go there, and aspartate gets put there. What is that genetic condition? Anybody know? That is sickle cell anemia. So I'm just illustrating for you the importance of how this system works in building proteins in our body. The order of the letters matters. So let's finish with this, and I'll introduce histo to you, and Monday I can start the histology. All right. So this is a random set of letters, LOCCKSR. What I'd like you to do for just the next 4 or 5 minutes is to think with me about these letters as letters in the English language, but used in an analogous way for comparing this to letters that I would have in the DNA molecule. So I'm going to try to argue, as we finish class today, an argument that was first made popular by Francis Collins, who is the current head of the National Institutes of Health. He wrote a book called The Language of God. He was in charge of the government's side of the Human Genome Project that was completed in the year 2000. He is arguably the most famous geneticist in the country. So this is the case that he made. He said that the TACCCGAG on the sense strands of the DNA molecule are in fact a language that the cell understands and interprets. So for him to say that the process of DNA to RNA is a transcription event is a language matter. The cell is copying the letters. Now it's an event where the T is copied as an A and the A is copied as a U. So it's not a perfect illustration, but it is a copy. It's a chemical copy. And then this process of the messenger RNA giving instructions for the amino acid is called translation. He's saying that's where the language really makes its major shift. Now the AUG is going to be translated in the cell as methionine. Brand new language. And he called this, in his book, the language of God. This is what he said from the podium when he announced it, which I found fascinating. He said, "Mr. President, we have discovered the language of God." Now, he didn't mean exactly what I'm going to show you, but I think he's right. So when we look at this here, when we look at this base structure of DNA, we can see that there's a G here and a C here and an A here. And everybody goes, yeah, I got you. These are triplets. They give message for codons. They give message for amino acids. And so if we take this one step further and we start asking fundamental questions of life, here's an important one. First, does it matter that this is a G? Could it be a C or an A or a T? Does it matter? Yeah, if you make a mistake, you get big problems. You can't just have random letters here. Okay? This is a language. Now, here's the second question. What dictates that this is a G? Now, if I unravel it in replication or in transcription, I know what dictates that this will be a C. Right? The cytosine nucleotide will come into the guanine. This is what they do. They attract each other. That is not the fundamental question I'm asking. The fundamental question I'm asking is, this is a DNA molecule in the cell. Why is G here and C here in the original? If it matters, then ultimately science needs to try to explain why G is here and C is here. So where are we going to look? This is what makes that decision. This bond between deoxyribose and the base is the one that makes the decision. So the question then becomes, we're just moving it further down, fundamental. The question then becomes, in this molecule right here, this deoxyribose, is there something specific, some nature about this glycosidic bond right here that dictates this will be an adenine and not a guanine or a cytosine or a thymine? And the answer is, there is nothing specific about the glycosidic bond. It doesn't care. You can put any base there in the construction of the original nucleotide. The chemistry will never be able to explain why this is adenine and not cytosine or thymine or guanine. We know what this bond is. It's nonspecific. Now, when Dawkins was presented with this, one of the most famous atheists in the world today, he's written several books. The God Delusion is probably the most famous of his books. When he was presented with this, he said, okay, I will grant you. Somebody wrote this language. But it had to be some kind of alien race that wrote the language somewhere else. And that alien race must have evolved wherever they were. Now, there's no way to prove him wrong in that. But I find it very interesting when you get to the bottom bond of life, the glycosidic bond to the base. The explanation, the only explanation in science today, is that something did this. So what we're left with, young people, is a decision about whether there is in fact a creator that wrote this language, or do you want to cast this out into deep space? You think that's comical, okay, when I tell people this, but Francis Crick wrote some very significant papers, and when I first started teaching at Louisiana College, the freshman textbooks had it in there, a concept known as panspermia, that suggested that aliens seeded life on this planet. Now, why would the guy who discovered the DNA double helix come up with such nonsense and put it in a science textbook as though it's science? Why? Because he knew a long time ago that naturalism, an atheistic point of view, would never be able to explain the glycosidic bond between deoxyribose and the base. The big problem. So I illustrate it this way. It's a language, you say. This is a language here, but it's nonsense, right? The language in the cell is certainly not nonsense. So I'll illustrate it for you this way. What if the letters that I just showed you on this page were in fact letters that were translated from 3-letter sequences? A very sophisticated language system, like codons, where GGC coded for C and UCC coded for L and AUC coded for K. And then I wrote the language this way. Instead of writing it this way, I wrote it with the codes. And I ask you to tell me what it is I'm saying here. Can you guys code this language for me? What does this say? You see, it's language. What does the language say? It's not nonsense. I have written a message. I have written you a biological message. The language of Wade. You're supposed to smile on that one. Not the language of God. The language of Wade. What did I say? I did. That's what I said. LC rocks. Yes? I've said something now. I didn't say nonsense. The language of the cell is not nonsense. It says something. It's language. Okay. When we begin class on Monday, tissues. We will start with epithelial. I promised you this in lab yesterday. So here it comes, starting on Monday.